Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series will include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. My name is Lynn Cox, and I'm a biochemist at the University of Oxford, studying the molecular basis of aging. I have a strong interest in discovering new targets and therapeutics for age-related diseases, and I serve on the clinical and translational theme panel of the Biochemical Society. Today's webinar is called The Biopharma Drug Development Pathway, Origins and Comparison with Small Molecule Discovery. And today we welcome Professor Alex Breeze from the Astbury Centre for Structural Molecular Biology at the University of Leeds, who also previously held a position for a number of years as Principal Scientist in Discovery Sciences at AstraZeneca R&D in the UK. In this webinar, Alex will provide a historical context for the development of modern biopharmaceutical drug discovery pathways. He will compare and contrast the evolution of biologics, that's large molecules development, with that of traditional small molecule pharma R&D, from which the biopharmaceuticals processes have largely been modelled and adapted. He will also explore the scientific and clinical challenges, attrition and risk profiles, economic drivers, and licensing approaches across the key stages in discovery and development for the two contrasting modalities of therapeutic agent. Along the way, Alex will touch on the factors behind the rapid rise in prominence of biopharmaceuticals, as well as the reasons that, despite the apparent conquest by biologicals, small molecule therapeutics will remain an important tool in the clinician's armoury. Questions will be asked at the end of the webinar, but you can send your questions in during the talk, and that will be quite helpful to us. If you have a question, please type it into the question box as shown in the image on the screen. I'll now hand over to Alex. We're very much looking forward to hearing your talk, Alex. Thank you.
Okay, so I hope everyone can see the screen. Just to give an outline of the webinar today, I'm going to be covering the historical context in which biopharmaceuticals development has evolved, and in comparison with the way classical small molecule drug discovery pathways have developed, to give a historical context for biopharmaceuticals. So we'll look at compare the drug development pathways for biopharma and small molecules, and look at the similarities and differences between small and large molecule pathways. And then we'll also look at comparative financial drivers, the finances of drug discovery, look at how small and large molecules compare in that field, look at the different attrition rates across the pharmaceutical development pathway for small and large molecules and the reasons for failure. So let's start off by having a look at some sales figures for Blockbuster pharmaceuticals, biopharmaceuticals in 2019. So these are biopharma drugs, and you can see that a lot of these, principally monoclonal antibody drugs, are really big selling drugs into the multi-billion-dollar sales annually. So drugs like Humira, Keytruda, Avastin, Herceptin, they're well-known biopharmaceutical drugs that have really come on the scene over the last 10, 15 years or so. And it would be easy to take from this slide the impression that sort of biopharmaceutical drugs are really sweeping all before them, and that maybe they are overshadowing small molecules as drugs. But let's actually look at where small molecules come in the list. So if we reinsert the small molecules into this sales table, we see that actually there are still a significant number of small molecule, traditional small molecule drugs that themselves are really very big sellers. So drugs like Eliquis, Revlimid, and so on, anticoagulants, treatments for multiple myeloma, tyrosine kinase inhibitors for cancers. These are all big selling drugs in their own right, and they're traditional, if you like, small molecules. So let's go back and look at, what we could consider to be the origins of modern drug discovery. And I guess we could consider, we could choose a number of different points in time for this origin of modern drug discovery. But my personal view is that there's no better place to start than with Paul Ehrlich, who was operating around the late 19th, early 20th century, and could be considered the founding figure of what he referred to as chemotherapy, which is what we really now call modern drug discovery. And so Paul Ehrlich originated the concept, which is now current, of screening compounds. And we now screen very large libraries of compounds in small molecule drug discovery for biological activity against usually a protein target, which is associated with or responsible for a disease process. And so Paul Ehrlich also contributed to developing what we now know as modern pharmacology in the form of the ligand receptor theory. And he was also responsible for coining the phrase a magic bullet. This is kind of an ideal drug, if you like, an ideal therapeutic agent that hits only the biological target that we're trying to inhibit to achieve a therapeutic endpoint. And so in many ways, I think Paul Ehrlich can be considered a significant founding figure operating around the turn of the 20th century. And so let's look at what the traditional small molecule drug discovery that's evolved from some of the concepts that Paul Ehrlich founded now looks like. And we can see that it's divided into essentially two major phases. There's what we call the discovery phase in the early part, and then the clinical development phase towards the latter stages. And the whole process is a long, complex process. It can take over 10 years to complete, from initially identifying a biological target associated with the disease process to approval and marketing of a drug into clinical availability. And it's expensive. It can cost on average between $1 and $5 billion to bring a new medicine to market. And it's very risky and very difficult. And to give you a measure of how risky it is, most drug discovery projects fail. Of every 100 or so drug research projects that are started at the beginning of the pipeline on the left-hand side, in terms of identifying a potential therapeutic target, only one of those drug projects will succeed and make it to the market as an approved drug. And I think this is brought even more starkly home if we look at the investment return profile of a drug discovery project. And so, if we look at the discovery and development phases, during those phases where we have research phase, preclinical and clinical development phases, for most of its life cycle, this is around about 10 years or so, a drug project is making a loss. So money is being plowed into R&D of the drug. And typical drug projects don't file patents until about the mid-discovery phase, but that's very early on in the drug discovery cycle, the project life cycle. And what it means is that the clock is ticking on that patent filing from really very early on in the drug discovery life cycle. So a typical patent life is around 20 years or so. And given that the drug project, if it succeeds to market, will take around 10 years to launch, then there's typically 10 years or less in which the drug project can actually recoup the investment that was made in it during the discovery and development phase, and start to make a return on that investment, profits that can then be plowed back into further R&D. And the limiting factor at the end of its patent life is what's termed the patent cliff. This is when other manufacturers can start to make generic copies of the drug, and that substantially undermines the revenues of the drug. So let's have a look at the earlier phase of small molecule drug discovery in a little bit more detail. And so discovery phase is divided up into a number of phases. So target identification and validation is the initial phase, taking about a couple of years or so. And this is really about establishing the link between biological target and the disease process. It's about developing biomarkers, developing assays with which to screen compound libraries, and these compound libraries can be up to 1 million or so compounds, developing in silico screening methods to complement those in vitro methods. And then we move on then to the hit identification phase, when, for example, a high throughput screen of up to 1 million or so compounds might be run, or the project might elect to do a what we call a fragment screen of smaller compounds, smaller numbers of smaller compounds, which have higher chances of producing a hit. When we have hits, there's a hit confirmation phase, developing initial structure-activity relationships between those molecules in the initial hit series, and starting to look at toxicology. Then selecting the best of those series, move into lead generation and optimization, further develop structure-activity relationships, start to optimize potency through targeted medicinal chemistry design make test cycles, and then look at pharmacokinetics studies, toxicity studies, start to think about filing in intellectual property patent filing at that point. And then the project, if it succeeded that far, moves into preclinical animal testing. So the questions that will be being asked by the project team during this early phase of small molecule drug discovery will be questions like, is the disease sector commercially viable? In other words, if $1 billion or so or more dollars are going to be plowed into the R&D on this project, does the project stand a chance of making a return on that investment? Does the target have a strong enough linkage to disease and essentiality in that disease? So if we hit the target, we effectively inhibit, and we're usually talking about inhibition of targets, although sometimes activation of targets is appropriate. But if we hit the target hard enough, is it going to produce a clinically meaningful response? And an important question in small molecule drug discovery is, can the target be therapeutically modulated with a small molecule? And a question that's often asked is this target druggable or ligandable? These are terms that will be heard in connection with the ability to therapeutically modulate a target with a small molecule. So let's look at some typical small molecule drugs and see what we mean by small molecule drugs. So here are some well-known examples of small molecule drugs. So ibuprofen is a well-known analgesic, cimetidine, a histamine H2 antagonist for stomach ulcers and heartburn. Down on the bottom left, we have vemurafenib, which is a relatively recent compound, a tyrosine kinase inhibitor, inhibitor of BRAF for malignant melanoma, and the well-known drug tamoxifen, estrogen receptor antagonist for gold standard breast cancer drug. So these are typical small molecule drugs. And let's have a look at some of the features that they share in common, some common characteristics of small molecules. So they are typically synthetic organic molecules manufactured using synthetic chemistry routes, containing carbon, nitrogen, oxygen, hydrogen, and occasionally halogens and sulfur. Small molecule drugs often mimic the effect of or inhibit the effect of endogenous ligands. So, for example, kinase inhibitors inhibit the binding of ATP to the kinase active site, inhibit the binding of the cofactor. Or they maybe inhibit binding to steroid receptors, for example. They may mimic the effect of natural toxins. The small molecule drugs usually comply with what have become known as the Lipinski rules, the Lipinski rules of 5, which are rather than rules, they're really a set of guidelines, which are taken to influence the likelihood of obtaining oral bioavailability for small molecule drug candidates. And so they look at things like molecular weight, number of hydrogen bond donors and acceptors, lipophilicity, and so on. And small molecule drugs are usually in the range of around 250 to 500 Daltons molecular weight. Okay, so we now have an idea of what small molecule drugs are like. Against that context, what do we mean by biopharmaceuticals? And I think we can draw perhaps a useful distinction between classical small molecule agents and biopharmaceutical agents if we look at really how they're made, how the bulk drug is produced. And if the bulk drug is made in a test tube in vitro using synthetic approaches, then we can consider it a small molecule drug. Often the number of synthetic peptides or oligonucleotides may be made through synthetic routes. On the other hand, if a therapeutic agent is made in a cell or an organism, I think that's a useful way of classifying it as a biologic or biopharmaceutical. That includes recombinant biopharmaceuticals such as monoclonal antibodies, and we can sort of loosely term these large molecule agents. And so here's an example of a large molecule biopharmaceutical or biologic. So this is pembrolizumab, marketed as Keytruda, which is an anti-PD-1 monoclonal antibody. And in common with other monoclonal antibodies, typically it's around about 150 kilodaltons in molecular weight, so vastly bigger and more complex than a typical small molecule drug. So let's then compare the early phases of biologics drug discovery with what we just saw of the small molecule process that has evolved over so many years. And if we look at the early phase of biologics drug discovery, you can see that there are actually quite a lot of common features here, but there are some differences. And so the start point is the same. It's essentially identifying a target with that has a biological activity that's linked to the disease process that we're interested in therapeutically modulating. So establishing that link, establishing biomarkers, starting to develop assays and routes for generating, for example, monoclonal antibody via inoculation with an antigen, for example. Moving on to the lead generation phase, choosing the modality that we're going to use, whether it's a monoclonal antibody, a full monoclonal antibody, or whether we might choose a different modality, such as a camelid variable heavy chain agent, setting up assays to test efficacy, so ELISAs, enzyme-linked immunosorbent assays, proof of principle efficacy assays, and setting up processes for in vivo pharmacodynamics and pharmacokinetic models. Moving on to lead optimization, assuming we get good start points, designing processes such as designing mutant libraries for humanization and affinity maturation, so developing the affinity further into the picomolar KD range ideally, looking at optimizing solubility and stability and engineering the FC region. And again, similar to small molecule drugs, starting to think about filing for intellectual property at this point, before then moving into candidate selection, so kind of the equivalent of preclinical studies for small molecules. And again, very similar questions are going to be being asked during this early phase of biologics drug discovery. So around commercial viability, the strength of the disease linkage the target. And an important question for biologics drug discovery is the question of whether the target can be therapeutically modulated with a biologic. And principally the question there may be whether the target is accessible to the circulation. So a very important question for a drug discovery team or drug discovery organization to be asking really in these very early phases is actually which route should we be going down? Should we be going down the large molecule route or the small molecule route? Which is going to be the better route for this particular target? And often that does actually depend on the nature of the target and particularly on where it's located within the body. So a couple of examples: if the target is the protein-protein interaction, so inhibiting the interaction of a protein target with, say, a growth factor. So, an example of that would be a receptor tyrosine kinase binding its extracellular growth factor. In a case like that, then it may well be that the more optimal route will be to go down a large molecule drug discovery approach, attempt to modulate this target with a biologic. And the reason for that is that protein-protein interactions typically have large binding epitopes, which are often hard to interrupt with a small molecule agent. So protein-protein interactions are a good class of target type where large molecules, biologics, often stand a better chance of success. On the other hand, if the biological target is an intracellular target, then it's rather more likely that we're going to need to take a small molecule approach to addressing this target. And the major reason for that is that we need cell membrane penetration in order to access an intracellular target. And cell membrane penetration is much easier to attain with a small molecule agent than with a large molecule agent. Of course, if we have an intracellular protein-protein interaction, then these are some of the more challenging types of drug target for the industry, and these are still remain very, very challenging. But really, it's about the addressing this question about the druggability or ligandability of the target with respect to small or large molecule approaches. So let's have a look at looking at comparing some features and characteristics of large and small molecule drugs. And I don't propose to go into this table in detail, but just to pick out a few key points. So in terms of target affinity and selectivity, biologics, monoclonal antibodies often have an advantage there. So they typically display very high target affinities, very high target selectivity, and that often translates to fewer off-target effects of biologics compared with small molecules. Biologics, in contrast to small molecules, are usually rarely orally bioavailable. So biologics are usually administered parenterally by injection or by inhalation, whereas often an aim for small molecule drug discovery programs is to have an orally bioavailable agent in the form of a tablet that's very easy to administer. So small molecule drugs can be self-administered, whereas large molecule drugs often require hospital administration. If we look at the lengths of the discovery phases, so small molecules typically have a longer discovery phase and a high attrition rate within the discovery phase. Biologics typically have a quicker discovery phase and generally lower levels of attrition throughout discovery and development. Manufacturing costs. There's a contrast between small and large molecules. So typically small molecules are relatively cheap to manufacture once synthetic routes for bulk, for scale up have been established. Scale up and bulk manufacture of biologics are typically a lot more challenging and significantly more expensive. And so a key feature of small molecule drug discovery, a really pivotal part of the process, is the preclinical phase where efficacy, toxicity, and DMPK profiling are carried out. So DMPK stands for drug metabolism and pharmacokinetics. And so this preclinical in vitro pharmacology and in vivo study of pharmacodynamics and pharmacokinetics are key features of small molecule drug development. So pharmacodynamics can be regarded as what the drug does to the body, if you like. Pharmacokinetics as what the body does to the drug, so how it metabolizes the drug. And a key aspect of small molecule preclinical work is safety pharmacology. So looking at human liver microsome studies and looking at metabolism by human liver microsomes and animal-based adverse effect studies. And then in preclinical studies in animals, looking at toxicological studies in at least 2 species, typically taken from rat, dog, or monkey species, and looking at animal and human biomarker studies. And these small molecule efficacy and tox profiling studies are really very, very important components of the small molecule process, taking place largely in the preclinical stage. These particular stages are probably slightly less of a challenge for large molecules, or differ in detail compared with small molecules. And let's have a look at some of the ways in which toxicity profiling differs between small and large molecules. So as discussed on the previous table, we see that target selectivity is more of a challenge to achieve with small molecules. So we typically get more off-target effects with small molecules. Whereas with large molecules, with biologics, we have fewer off-target effects. And that means that generally target-associated or off-target associated toxicity rather is less of an issue with biologics than with small molecules. So that does reduce the impact of one of the features of tox studies in biologics development compared with small molecules. But on the other hand, species selectivity of small molecules is typically broad, so that that opens the way for toxicity testing in a number of different species, animal species, before moving into human trials. Because biologics often have higher species selectivity, this can create a bit more of a challenge for preclinical testing for biologics. If we look at half-lives, small molecules have short half-lives. They're rapidly metabolized and excreted. Biologics have typically longer half-lives, often in the range of days, and that does entail some precaution in developing clinical trial protocols. Small molecules have large volumes of distribution, whereas biologics typically have much smaller volumes of distribution, in that they're restricted to the vascular system. So we have plasma concentrations of biologics, but they're not generally widely distributed throughout tissues. I guess probably the key difference between small molecule and biologics preclinical testing is the question of immunogenicity. And small molecules typically have relatively little immunogenicity, and this is a relatively minor consideration. But for biologics development, this is really a major consideration in developing testing protocols. It's a common challenge to achieve a biologic that has a minimal immunogenicity profile, immunogenicity risk. So let's move on and look at clinical development. Assuming we've got our agent successfully through the preclinical development phases, we're ready to start clinical trials in humans. These are separated into 3 phases, typically. Phase 1, where we're looking at a small number of human volunteers, maybe up to 50 people, healthy volunteers, and really the aim here is to establish safety, an early signal for safety of the agent. Moving into phase 2, assuming we pass phase 1 successfully, we start to recruit more volunteers to the trial, and these volunteers will now start to include patients. And here we want to start to get a signal for not only safety but also efficacy. So does the therapeutic actually look to be successfully modulating the disease process? And if we get through phase 2 successfully, we then move into much larger studies in phase 3, where we recruit up to several thousand patients to really get a much firmer grip on the questions of efficacy and safety. And those phases the phase 3 can take anything up to several years. And so we have typically up to 6 or so years or more in the clinical development phase. The differences actually between small and large molecules in the clinical development phase is actually not so great, and they mainly relate to the routes of administration. So small molecule agents will typically be orally bioavailable agents and administered once or twice daily in tablet form, whereas biologics typically are parenterally administered by injection and maybe much less frequent dosing. So the routes of administration, and also with large molecule drug development and clinical trials, are really a very strong focus, particularly in the early phases, on any signs of immunogenicity and adverse immunogenic reactions. So let's have a quick look then, moving on at the licensing processes for small and large molecules. And I'm just citing here the reference process from the US Food and Drug Administration, the FDA, which is really, has formed largely the blueprint for most regulatory processes across the world in most jurisdictions. And if we look at the small molecule licensing process, typically this will comprise 2 to 3 phases in a drug molecule's life cycle. So going from the preclinical into the clinical trial phases, there'll be an application for an investigational new drug with what we would then call at that point, with a molecule having successfully completed preclinical development, a new chemical entity, an NCE. Then if that compound successfully gets through phases 1, 2, and 3 of the human clinical trials, then the next stage is to file a new drug application with the FDA with what we now will call a new medical entity, an NME, to seek approval from the licensing agent for marketing of that drug. And then, assuming that approval is granted and the drug is then marketed, it will have a certain number of years of its patent life remaining before that patent then expires, and other companies can then start to come in with generic versions of that drug. And at that point, they will need to submit what we call an abbreviated new drug application, an ANDA, for approval of their generic version of that drug. For protein-based drugs, large molecule therapeutic development processes essentially developed a set of processes that follow rather closely the small molecule processes that have been developed by the FDA and by other agencies. So we'll have new biological entities going into clinical development, which will then, at the end of a successful clinical development, result in a biologics license application for a new biologic. And then similarly, following a number of years of marketing, assuming approval of that biologics for sale on the market, and these we will then get a process for competitors to start to come in with copies of that biologic drug. And this process for what are now called biosimilars which are the biologic equivalent of generics in small molecules, is really still under development, and I think still subject to refinement, but we largely have a process in place now for biosimilar or follow-on biologic approval and generation of an abbreviated biologic license application to approve that generic version of the biological drug. So let's have a look at some comparisons between small molecule generics and large molecule biosimilars. So the generic is really a copy, if you like, of the original small molecule drug that was licensed by the licensing authority. And it's really an exactly identical copy of the small molecule drug. So the costs to generic manufacturers are really much lower than the costs to the original innovator of the drug, because the innovator's already paid the discovery and development costs. And the costs to manufacture a small molecule are, relatively speaking, quite cheap. So the generic manufacturer really has only to establish a process for bulk manufacture of the drug and tableting the drug and applying for the generic license for that drug. And that's a relatively inexpensive process for the generic manufacturer. Biosimilars have some similarities to generics, but also some important differences. So biosimilar is the biological equivalent of a generic small molecule drug. And a biosimilar really needs to demonstrate that, not that it's identical to the original biological drug, but merely that it's highly similar to the reference biologic that's been approved by the licensing agent. And it also has to demonstrate that it has no clinically meaningful differences from the reference biologic. And this is a reflection of the fact that actually it's much more difficult to produce an absolutely identical agent as a biological. And that something that's similar enough, both in structure and in clinical outcome, to the original biologic is in principle approvable as a biosimilar. So biosimilars cost significantly more to produce, to develop and license than generics. So they may not have the same profit margin, but nonetheless there is still a significant incentive to companies to produce biosimilar versions of biopharmaceuticals. So let's compare the economics of small molecules and biologics. So small molecule development costs are usually lower on a per project basis than the development costs of large molecules, biologics. And that's largely down to the higher cost of the development phase and the manufacturing phase of biologics development. But also a key contribution to that is that the attrition rate of small molecules is significantly higher across all the phases. Biological drugs, biologics, for example, monoclonal antibodies, typically have higher return per patient. So the cost of biological agents is often in excess of $10,000 per patient per year, and that reflects the high cost of development and manufacture of the biologic. A corollary of that, of course, is that the high cost per patient per year of biologics can be a barrier to wider patient access compared with a small molecule. And we often find that small molecules that are achieving sales volumes of comparable to those of large molecules actually are doing so by treating much larger numbers of patients compared with the equivalently grossing biologic. Biologics have, as I've mentioned already, have significantly lower attrition throughout all the phases of discovery and development. And that means they have a higher overall probability of success. And this is a key, I think, key attribute as one of the attributes that's driven the advent of biologics as a major player in biopharmaceuticals and clinical therapeutic agents. And this is illustrated in this graph here, where we can see that the probability of success really across all phases of development is higher for biologics than for small molecules. The difference is highest in the early stage of clinical development, so first human dose to market, the phase 1 clinical trials and beyond. The success rate advantage, probability of success advantage for biotech agents, biologics, is really significantly ahead of that of small molecule agents. As we move through clinical development, then the advantage is eroded to a degree, but still biologics have a higher probability of success, even at submission for approval to market. So this is my last slide to wrap up and to conclude then this comparison of small and large molecule biopharmaceutical development pathways. So large molecule biologics drug discovery and development has evolved from, has been informed by the established processes established over many decades for traditional small molecule pharmaceuticals. There are high degrees of complementarity between small and large molecule drugs. Both modalities still have a very prominent and important role to play in pharmaceutical development, in developing effective clinical agents to meet unmet medical need. And importantly, they present different clinical and financial risk-benefit profiles. It's very important for teams developing therapeutic agents to weigh up the advantages and disadvantages of small molecule versus large molecule approaches for the particular therapeutic target that they're aiming to address. And I think the last comment with which I would wrap up is that despite the continuing rise of biopharmaceutical products and the apparent inexorable conquest of the pharmaceutical market by biological agents, small molecules continue to play a very strong role and will continue in the future to play a very strong role in treating unmet medical need, and will continue to be highly complementary to large molecule drugs. So I'll wrap up there, and I'd like to thank you very much for your attention, and obviously we'll be very happy to answer any questions you may have.
Hello, Alex, that was fantastic. Now, I'm not sure if you can actually see me. We appear to be having some issues with camera still, but I'd like to start with some questions from the audience. There's been a few very interesting questions coming in. Jessica asks, a very telling question based on what you were saying about accessibility of biologics versus small molecules. Which approach would you take to target intracellular protein-protein interactions?
Well, as I mentioned, this is an extremely challenging area still, and there have been some successes with small molecules for intracellular protein-protein interactions. So, for example, BCL2, BCLXL inhibitors, navitoclax, and compounds of that nature are an example of successful agents, small molecule agents targeting intracellular protein-protein interactions. But they do remain very, very challenging for small molecules, but of course, almost equally challenging for large molecule large molecule therapeutics, where the challenge is actually for those large molecule agents to cross the cell membrane and gain access to the target. Of course, there are other approaches, modalities such as mRNA-based approaches and so on, that are also potentially proving successful and may prove to be equally promising routes forward for intracellular protein-protein interactions. So I think the jury's still out on what's going to be the best approach, and it may well be that I think it's going to be hard to generalize, and teams, drug discovery teams targeting intracellular protein-protein interactions are probably going to have to take a lot of different shots on goal to tackle these targets effectively.
Yeah, so following up on that, there's the issue with antibody access to tumours. So you were talking about the distribution of these drugs and the fact that biologics are generally restricted to vascular distribution. And yet when you're trying to target tumours, quite often they're poorly vascularized. So how do you ensure that you can get access of biologics into the middle of a tumour?
Well, yes, I mean that is a significant problem, and it is a significant factor that actually can limit efficacy of biologics targeting tumors. And I think particularly in the field of oncology, actually I think what we're going to see increasingly is that combining different modalities is increasingly going to be the most successful way forward, both from the point of view of addressing questions of accessibility of the target to different agents, whether it's small or large molecules, but also in terms of addressing development of resistance, which we know is a major issue in developing cancer therapeutics.
Mm-hmm. Now, David Wood comes up with a very good point. In your timelines, you showed that the early discovery stages in small molecules was a couple of years, and in biologics, it was quite significantly longer. So for optimal discovery and development of new drugs, should the lengths of patents for small molecules and biologics be the same? And is there any case to revise how patents work?
Well, that's an interesting question, and I mean, I think the lengths of those phases are not that different in terms of the early phases. I mean, typically, actually, the early discovery phases for biologics are probably, if anything, slightly shorter than for small molecule agents. But I mean, I think this is a question that's actually continually being revisited about patent lifetimes, and whether there are cases to be made for extending patent lives on pharmaceutical agents, or altering them in some way, and/or what the optimum time for filing for intellectual property is. And I think, I mean, there's no one answer to this problem. I think, I mean, there has been a trend in maybe in small molecule discovery to file a little bit earlier, but then I think generally, we often see that these trends sort of oscillate from, around a median point over a number of years. Okay. I think it's difficult to provide an answer to that.
No, I know, but it's one of those bugbears, isn't it? If you file too early, then you don't have the patent lifetime. If you file later on, then there are consequence issues on that as well. So moving to something slightly different, Catherine has pointed out, obviously, that so many of the small molecules fail. And as far as I'm aware, they tend to fail in early stage clinical trials, even though they've shown good efficacy in preclinical studies. So Catherine asks, what are the common properties or reasons of drug molecules that fail during clinical trials?
Okay, well, that's a very good question. And well, I don't know whether a common problem. I mean, certainly, I mean, I think there's been a lot of work done in terms of, looking at factors that influence attrition in small molecule drug discovery over the past 20 years or so. And so, focuses on things like, for example, lipophilicity, the biophysical properties of small molecules, lipophilicity, molecular weight, and so on. These often tend to be negatively correlated with success. But again, it's very hard to generalize in those aspects. A lot of it is not necessarily related to the particular molecule, but related to the strength of the hypothesis around the link of the target to the disease. And so, I mean, there's a lot of effort going into sort of actually more front-loading more the target validation stage and getting more early signals of clinical relevance of the modulating the target. So that I think it's a combination of both addressing issues to do with the properties of the small molecules themselves, but also actually learning a lot more earlier on about the link of the target to the disease.
Yeah, I mean, there's a follow-up on that from Lauren, which I think is a very insightful question. So she's asking about improving the success, moving from preclinical to clinical, and perhaps considering emerging importance of things like the microbiome. So that presumably can have a huge impact on how a drug is metabolized, the interactions within the body, the targets. So how do we take that into account, given the very formulaic way that preclinical studies are conducted?
Well, yeah, that's a very good question, and I think it is undoubtedly a neglected area. I mean, I think particularly in small molecule discovery, I think the microbiome can have a very large influence on some of the aspects of preclinical, the preclinical aspects in terms of drug metabolism, pharmacokinetics, and toxicity, and I think as an industry, I think the industry will need to pay significantly more attention to that. I think it is probably an under-investigated area, and I think we will see more attention paid to the role of the microbiome in the success or otherwise of small molecule agents.
Okay, thank you. Now, Jack asked a very pertinent question given the COVID pandemic. There's obviously been warp speed pushing to get a vaccine out in the States incredibly quickly, and also around the world. How do companies and projects get to later stages quicker? How can we speed up the whole process, either of vaccine or drug discovery?
Well, yeah, that's a very interesting question because I mean, clearly, the process, the processes have evolved to very carefully monitor and come to a view on efficacy and safety, and there is certainly a limit to the extent to which those processes of clinical development can be short-circuited. And a lot of it actually is about maintaining, particularly in the context of COVID vaccines, actually is about being able to maintain public confidence in the product, and to ensure that the public is confident that the usual processes which have been put in place over many decades to ensure that agents meet the requisite safety profile and are sufficiently efficacious to warrant bringing them to market at what are often premium prices.
I think
the public actually has confidence in that process, and the confidence of the public isn't undermined by short-circuiting the processes. But having said that, clearly there are opportunities to streamline some of these steps, and we are seeing actually the extent to which that may be possible with the development of COVID vaccines, and how to what extent these are translatable to to the broader context of biopharmaceuticals development in the post-COVID world. I think will remain to be seen. It'll be really interesting to see what lessons can be learned from this and can be applied more generally to pharmaceuticals development.
It's very interesting that you point out public confidence there, because obviously there's a concern of over-promise and over-hype on the vaccine, and perhaps we need public education that even a very good vaccine could potentially only protect 50% of the population. So yeah, it's all about being open about the whole process.
With and being open with the data, and actually being open with the nuance of the data, and actually ensuring that the public appreciates that there's nothing's black and white here. I think is going to be very important.
Yeah, I couldn't agree more. I mean, we've got to maintain faith in, well, confidence in science. Now, Gemma asked a question about development of biologics and how do you manipulate biologics to reduce the likelihood of having an immune reaction? Can you predict how immunogenic a biologic would be?
Yes, I don't know the answer to that question, I have to confess. I mean, I think there are a lot of processes that have been established, well-established process of humanization of antibodies, which can certainly minimize the risk of immunogenicity. But I'm not an expert in that field, and I don't know what the latest developments are in that area in terms of predicting the immunogenicity of biologics.
Mm-hmm. Now, as we're running close to time, I'm going to have to wrap it up there. But for anyone who's interested, and for those of you, sorry, who we didn't have a chance to get your questions answered, you can continue the conversation online. You can see on the screen. So you can follow at BioChemSoc and at PPP Publishing on Twitter, or use the hashtag Biochem Online and Biochemistry Focus. So thank you, Alex, for an excellent and enlightening seminar. And thank you to the audience for being such engaged participants. In these new and challenging times, it's more important than ever to stay connected. It might be an extraordinary time for everybody, but it's also an exciting time to become involved in the Biochemical Society as they look to expand their online activities. So do join the community of researchers and specialists to stay connected and take advantage of the key benefits, which include discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to a couple of journals, and more. So visit the website to find out more. I mean, I've found being a BioChemSoc member incredibly enlightening, and taking a more active part has been particularly useful in my career. So do note that there are a couple of posts being advertised at the moment for society. So do consider taking a more active role where you can drive the future of biochemistry and the future of the Biochemical Society. On that front, we would welcome your suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So if you have an idea for a webinar, we invite you to submit a proposal, and you can find more information about this on the website at www.biochemistry.org/webinars. So do join us for the next webinar in the series, also on the topic of drug discovery, and that's happening on the 30th of September at the slightly earlier time of 2 p.m. This will be presented by Sian Estelle from Covance Laboratories on a brief history of biopharmaceuticals, scientific drivers for adoption of large molecule therapeutics. And again, more details on this can be found on the website. And I'd just like to thank Alex one more time. That was a really interesting and enlightening talk that's put everything about drug discovery in a context for me to help me understand it much better. So thank you very much, and thank you to the Biochemical Society organisers who've made this happen. And finally, apologies that we couldn't get the tech working. There's some Mac issue on a camera, but I hope you could hear me clearly anyway. Thank you for your time and attention, and goodbye.